Hello, everyone. My name is Xian Wang. I am a postdoc researcher at the Hospital for Sick Children at Toronto, Ontario. I am the recipient of Down with Cancer Research Fellowship for 2021. It's my honor to present my project, Targeted Mechanical Ablation of Glioblastoma using spatial-temporally controlled magnetic carbon nanotubes. Glioblastoma, or GBM, is one of the most aggressive type of cancer that begins within the brain. Glioblastoma is known for its high fatality rate, high recurrence rate, and therapy resistance. The current treatment approaches for GBM include surgery, chemotherapy, and radiotherapy, but the 5-year survival rate for GBM patients is still very low. Since FDA's approval of using the chemotherapeutic agent temozolomide to treat GBM in 2005, all subsequent clinical trials that target various signaling molecules failed. A unifying resistance mechanism emerges. Surprising one or a few proteins or pathways give rise to an alternative, non-targeted biochemical signaling mechanism to facilitate GBM growth. A novel treatment approach without relying on a specific signaling pathway is desperately needed. Inspired by our previous works studying the mechanical properties and mechanosensation of brain tumors, I propose an out-of-box approach to physically destroy cancer cells using high-precision magnetic control and biocompatible magnetic carbon nanotubes. A carbon nanotube is a tube made of carbon with diameters typically in the scale of nanometer. With their small size and biocompatible carbon nanotubes have been applied in biomedical engineering as cell growth scaffold, drug carrier, contrast agent, and for cytoplasm viscosity measurement, and for cell membrane lysis. In my project, the magnetic carbon nanotubes were introduced into the glioblastoma stem cells and cultured with the carbon nanotubes for 24 hours. Then applied a rotating magnetic field to the whole petri dish for 30 minutes. The cell level that ratio were calculated through tryptophan blue staining assay. The results show that rotating magnetic field can actuate the carbon nanotubes and cause around 30% cell death. The magnetic carbon nanotubes show low cytotoxicity to the cells without magnetic field applied. I also studied how different magnetic field parameters would affect the treatment. The results showed that the magnetic field with 20 millitesla field strength and 20 Hertz rotating frequency caused the most cell death. The treatment was then applied to the treatment of GBM-bearing mice. The magnetic carbon nanotubes were introduced into the tumor 7 days after tumor implantation. Then 5 rounds of magnetic treatments were applied to the mice. The tumor size and animal body weight were observed during and after treatment. The treatment magnetic field was generated based on the magnetic field model we developed. The tumor position was first quantified in bioluminescent imaging, then transformed into the Cartesian coordinate system. The magnetic field was then applied so that the tumor region received magnetic field always larger than 20 millitesla throughout the treatment, while the surrounding healthy tissue received less dose of magnetic field treatment. The results show significantly prolonged survival of GBM-bearing mice after the treatment, as shown in this right curve. The median survival time increased from 22 days to 26 days. Through bioluminescent imaging, we observed less tumor burden in the treatment group compared to the control group, only receiving the magnetic field without carbon nanotube injected. Next, I am working towards the surface functionalization of the magnetic carbon nanotube with tumor-recognizing antibody. If successful, it will improve the treatment efficacy and treatment specificity towards the tumor, while causing less damage to the healthy brain tissue. As shown in this figure, the green signal indicates the tumor cells, and the red signal indicates the carbon nanotube we introduced. The preliminary functionalization of the carbon nanotube significantly increased the concentration of the carbon nanotube within the tumor region compared to the non-functionalized carbon nanotubes. In addition to functionalization, I'm also working on validating the proposed treatment on chemotherapy-resistant tumors. Hopefully, this approach will provide an alternative approach for the tumors currently untreatable. In the end, I would like to thank my colleagues and collaborators from the BrainTumour Research Center at the Hospital for Sick Children and the Engineering Department at the University of Toronto. Thank you for Done with Cancer and BrainTumour Foundation of Canada. Your support has made this work possible. If you have any questions for me, feel free to contact me through the BrainTumour Foundation of Canada. Thank you.